Welcome back, everybody, to Chapter 2 of the OpenStack Psychology textbook. My name is Prescott. I'm here with Succinct Psychology, and this is going to be a short video because we're just going to be finishing up Unit 2, talking about different research methods and experiment methods and problems, all that good stuff. So, without further ado, let's get going. Last video, talked about some issues to consider when conducting research, variables, selecting participants, placebos, designing your experiment, all that fun stuff. So, continuing on, we have interpreting experimental findings. So once data has been collected, a statistical analysis is conducted. So a statistical analysis determines how likely any difference between experimental groups is due to chance. Psychologists usually discuss results as significant. Oh wait, I went through this last video, but we'll keep going. Psychologists usually discuss results as significant or non-significant. If the odds that the differences occurred by chance are 5% or less, then the results are significant. A true experiment, including random assignment, manipulation of the independent variable, and control of extraneous variables, reduce the odds of results occurring by chance. So if you have all these things, you're really covering all your bases into making sure that your results are significant, or the odds that the differences occur by chance are 5% or less. So now, reporting findings. This is where we left off last time, my bad. So research is usually reported in scientific journals, which are usually aimed at an audience of professionals/scholars, and articles published are peer-reviewed journal articles. Peer-reviewed journal article is an article read by several other scientists, usually anonymously, with expertise in the subject matter who provide feedback regarding the quality of the manuscript before it is accepted for publication. So this helps to weed out poorly conceived or executed studies. It improves articles with suggested revisions and determines whether the research is described clearly enough to be replicated by other researchers. So replication determines reliability of the original research design, can include additional measures that expand on the original findings, and provide more evidence to support the original finding or to cast doubt on those findings. So replication is really important because it makes sure that you're not following experimenter bias, you're not just looking for certain answers. It allows other people to test out your research and see if they get similar results, and therefore make sure that it is significant. All right, so bad science and retraction, the vaccine-autism myth. So this is kind of going into how, being peer-reviewed can be a little skewed, which is important. Why replication is important, because you need to make sure that your results aren't skewed, they're not biased. So many peer-reviewed publications publish research making claims that routine childhood vaccines cause some children to develop autism. Since these reports, large-scale research was carried out suggesting that vaccinations are not responsible for causing autism. Many of the original studies have since been retracted. So it was found that the leading research in the original study had a financial interest in establishing a link between childhood vaccines and autism. Unfortunately, the initial claims were publicized, and many people still think vaccinations cause autism. So that's why replication is important, because they had to do research and make sure that these claims were valid when it turns out they were not. So, going on, we have reliability and validity. So reliability is the consistency and reproducibility of a given result. So would the same test give the same results every time? Or do the instruments/tools used to collect data do so in consistent, reproducible ways? When a study involves observations by multiple people, it is important that they all make observations and record them in the same way. So this is inter-rater reliability, which is a measure of agreement among observers on how they record and classify particular events. So inter-rater, in between raters, or observers. So a reliable, consistent measurement does not always mean that it is measuring something correctly. Validity is the accuracy of a given result in measuring what it's designed to measure. So does a test measure what it is meant to measure? Important thing to remember is a valid measure is always reliable, so it's consistent. But a reliable measure is not always valid. And a way to think about it is Think about, shooting arrows at a big target. So validity would be, your arrow's hitting the bullseye, like it's measuring what what you want it to. In this case, the arrow is hitting the bullseye. That's what you want. Reliability means that if you shot a bunch of arrows, they would be in a tight cluster because they're consistent. So you can think about that when thinking about research, reliably consistent or validly accurate. So is it measuring validity, measuring what it wants to, reliability, are they consistent? Are the results consistent? Next, we have ethics. So, research involving human participants. Research involving human participants must adhere to strict guidelines. So for that, we have the Institutional Review Board, or the IRB, which is a committee of administrators, scientists, and community members that reviews proposals for research involving human participants. So they exist at any research institution that receives federal support for research involving human participants. Generally, IRB must approve research proposal before it can proceed. So, important part of the IRB and ethics involved when you're doing research with human participants is informed consent. So it's the process of informing a research participant about what to expect during an experiment and then obtaining the person's consent to participate. This includes potential risks involved, implications of the research, notification that participation is voluntary, and a notification that any data collected will be kept confidential. So if you've ever done any kind of research, then they will have given you informed consent. They will have told you what the research is about, potential risks, that, if you want to opt out at any time you can. That's what informed consent is. So here we have, as institutions, IRB meets regularly. In institutions, IRB meets regularly to review experimental proposals that involve human participants. So if you want to do something with human participants, you have to run it by the IRB. So part of ethics, when it comes to research, is deception. Sometimes deception is necessary to prevent the participant's knowledge of the research question affecting the results as long as it is not considered harmful. That's a key point. So deception is purposely misleading experiment participants in order to maintain the integrity of the experiment. And debriefing is when an experiment involves deception, the participants are told complete and truthful information about the experiment at its conclusion. So if you're going to use deception, you have to use debriefing. You can't deceive somebody and then just not tell them. And an example of when researchers did this, they did not use debriefing, was the Tuskegee syphilis study. So ethical guidelines have not always existed, obviously. In 1932, participants were recruited in an experiment studying syphilis in black men. The participants that tested positive were not informed that they had the disease. Although no cure existed at the beginning of the study, a cure was found in 1947 in the form of penicillin, but it was not administered to participants. So many participants unknowingly spread the disease and many died. And that's because they weren't debriefed. They were deceived and they were also not debriefed, which is nowadays a huge violation of the IRB's ethics with human research subjects. And finally, with ethics, we have research involving animal subjects. So 90% of psychological research involving animal subjects uses rodents or birds. Animals make good substitutes because many of their basic processes are sufficiently similar to those in humans. Animals are used when the research would be unethical in human participants, and researchers must still aim to minimize pain or distress, so you can't go and torture cute little animals, thankfully. Institutional Animal Care and Use Committee, or the IACUC, is a committee of administrators, scientists, veterinarians, and community members that reviews proposals for research involving non-human animals. So rats, like the one shown here, often serve as subjects of animal research. So the IACUC is the animal version of the IRB, both with human research subjects and animal research subjects. There's ethical guidelines. You can't just do whatever you want to either. That's not ethical. And that concludes Unit 2 of the OpenStax Psychology textbook. I hope you guys found these videos very informative and that you continue to watch, because I'm really enjoying doing this. I hope you guys are enjoying following me along for the ride. Again, my name is Prescott. I'm here with Psychcinct Psychology, and I hope everybody has a great day, and I will see you next time. Bye-bye.